stop sharing.
Okay, I'm sharing my slides. Sounds good.
1 second. Yep, looks great. Okay, go ahead.
Thanks, Chen, for the nice introduction on the exciting seminar series. Thanks for having me, and thanks everyone for coming. Okay, so let's get started. Hi everyone, I'm Chenyi Tu, a NASA Postdoctoral Fellow at Duke University, working with Mike. Today I'm going to talk about the role of alkalinity in the origin of phosphate deposits. The evolution of the marine phosphorus cycle may have played a key role in shaping ocean atmosphere oxygen levels, alongside major episodes of biospheric innovation and radiation throughout Earth's 4 billion-year history. Reconstructing past marine phosphorus cycling is therefore critical and often relies on geochemical signatures preserved in ancient sedimentary rocks, including chemical precipitates, binding iron formations, and carbonate rocks, and fine-grained cystic sediments and shells. Among these, phosphorites stand out as a member archive of past phosphorus cycling. They are sedimentary rocks with exceptional phosphorus enrichments, rich in phosphate minerals such as apatite. Here we refer to rocks with at least several weight percent of phosphorus oxides. They are commonly interbedded with shales, limestones, dolomites, chert, and display various sedimentary textures, such as colloids, buoys, and clasts. Phosphorite deposition is frequently linked to shallow and sediment-stuffed environments, and they're commonly found in association with stromatolites within the same stratigraphic successions and with evaporative deposits within the same depositional basins. And deformation is influenced by sea level fluctuations with maximum phosphorus enrichment typically occurring during marine transgressions. However, there's an open debate about the dominant controls on phosphorite prevalence throughout Earth's history. One school of thought argued that phosphate occurrences reflect changes in global seawater nutrient inventory. The reason is phosphorites are globally distributed into notable intervals, the Paleoprozoic and the Neoprozoic Cambrian transition. Both periods were marked by tectonic upheaval, global carbon cycles, and increasing oxygen levels in atmosphere. For example, some models link the formation of Paleoprozoic phosphorites to intensified weathering and resulting increase in phosphorus fluxes to the oceans. And the other school of thought argued that phosphorite formation may have been driven by localized water mass chemistry within hydrologically semi-restricted basins. The reason is some phosphorus-rich sedimentary successions in Earth's history exhibit anomalous geochemical signatures, such as unusually high N15 values and carbonate N13C ratios. However, these data are difficult to reconcile with global marine signals. Recently, growing studies indicate that elevated N15N values in Asian rocks may reflect high pH and alkalinity, so-called carbonate-rich, in local aquatic environments. So analogs to modern aqueous systems, alkaline conditions are known to promote phosphate accumulation in the water column. If the accumulated phosphate reservoir was subject to rapid burial, this could lead to phosphorus enrichment in sediments. So based on this framework, we hypothesize that many phosphate deposits in the geological past could have been associated with hydrologically restricted settings where phosphogenesis was primarily controlled by local factors such as elevated alkalinity. Interestingly, the Eocene Green River Formation contains co-occurring alkaline minerals such as trona and nickelite, elevated N15N signatures, and episodic phosphorus enrichments, consistent with our hypothesis. So to further test our conceptual model, we focused on the Permian phosphorite formation, a phosphorus-rich sedimentary sequence deposited within a subtropical epicontinental sea the phosphorus sea along the western margin of Pangaea. And this unit represents one of the world's largest known phosphate rock resources. And phosphorites were deposited over a vast area of the western United States. Importantly, there is no direct modern depositional analog. And the phosphorite formation comprises diverse lithologies, including phosphorites, churs, and organic rich metastomes. You can see this stratigraphy column. And phosphorus-rich beds primarily concentrated in the mud peak and retort members. This succession deposited on a low-gradient carbonate ramp less than 200 meters, with phosphogenesis taking place both in basinal settings and shallower inner ramp environments. And there are two age models proposed. One constrains sedimentation from the latest Precambrian to the end of the Verdian, and the other extending deposition to the Permian-Triassic boundary. In addition to the phosphorite formation, other sedimentary sequences also accumulated in the phosphorite sea. You can see there are shallow marine CC classics, sandstorms shed from the Milk River uplift to the north, and carbonates are basically the Park City Formation, and also evaporate deposits, the Goose Egg Formation, the Highlight and gypsum, that are located in the desert to the east. The origin of the extensive phosphogenesis remains elusive. Most models propose an upwelling system along the western margin of Pangaea, analogs to modern settings such as Peruvian shelf margin. However, the phosphorite sea was at least partially isolated from open Peninsula Ocean by a remnant of the Antler Highland, and bordered to the east by arid, highly evaporative subhar environments. So based on this paleogeographic configuration, we hypothesize that this sequence may be instead deposited in a semi-restricted setting, where local factors could have played a significant role in phosphogenesis. And we have one geocore and two outcrop materials and sections. For the core, it was taken from western Wyoming, represents a western Wyoming capturing a nearly continuous section of the phosphorite formation, and representing a mid to inner ramp depositional setting. For the two outcrop sections, they are located near Idaho-Wyoming boundary, capturing part of the retort member and part of the mud peak member respectively. They represent similar depositional environments as the geocore. In this study, we use delta 15N major and trace elemental analysis to test the potential link between phosphorus enrichment and alkalinity. So we first categorize the chemical properties of the phosphorite basin. Unfortunately, most samples contain relatively low ion concentrations, less than 0.5%. So this precluding the use of ion speciation. Instead, we focused on redox-sensitive trace metals and TOC concentrations. So basically, the barrier rates of both trace metals and organic carbon are typically elevated in sediments deposited beneath reducing and euxinic bottom waters compared to well-oxygenated seawater. Okay, so now let's focus on our data. First, this column is phosphorus oxide contents. You can see they are episodic enrichments. Our geocore samples are generally categorized by relatively low TOC, less than 1%, and muted concentration of molybdenum and uranium, except in the lower portion of the section, and the phosphorus-rich samples for uranium. The latter makes sense because apatite is a major sink of uranium, so it has higher uranium concentrations. The pattern suggests that most samples were deposited beneath oxic to non-euxinic reducing bottom waters, consistent with the generally low organic carbon total phosphorus ratios observed in our samples. By contrast, several samples from the lower part of the section exhibit remarkably higher molybdenum concentrations. Unlike many other trace metals, molybdenum is quite sensitive to euxinic conditions. So our data suggests that in intervals of at least transient euxinia within the phosphorus basin. Additionally, our samples from euxinic interval exhibit relatively low molybdenum TOC ratios, less than 30. In sulfidic-rich settings, sedimentary molybdenum TOC ratios can be further used to constrain the degree of water mass restriction. Detailed studies on more than restricted basins show that increasing restriction is closely correlated to lower molybdenum TOC ratios recorded in sediments such as the Black Sea and Precambrian fluid. Two processes can lead to these muted values. One is rapid drawdown of trace metals from water column, and second is limited renewal, seawater renewal from open ocean. So in general, the molybdenum TOC ratios are comparable, of our samples are comparable to those of the modern Cariacos Basin, and lower than those of the euxinic settings more connected to the modern ocean, such as the Stanic Inlet. This data suggests that the phosphorus basin was semi-restricted. And this is supported by previously reported strontian isotope data. You can see they are more radiogenic compared to the coeval strontian isotopes of the global ocean. How about salinity of the water masses within the basin? We have a few observations. First, dolomites and extensive evaporate deposits, including anhydrite and halite, are documented within the basin. Climate simulations and geochemical data also suggest evaporative conditions. Moreover, elevated concentrations of gammazine indicate salinity stratification, provide additional support for salinity stratification. So all this evidence indicate that the water masses within the phosphorus basin were more saline than the normal seawater. Interestingly, we find exceptionally high delta 15N values greater than 15 per mil in both drill core and outcrop samples. You can see data here. And the bulk nitrogen isotopes closely match, and the bulk and kerogen nitrogen isotopes closely match the corresponding bulk nitrogen. And these values are markedly higher than the global average for modern marine sediments and other Permian strata. Before sorting out possible mechanisms of this trend, we have to first consider the potential effects of metamorphism. Numerous studies have shown that thermal duration can elevate delta 15N and carbon nitrogen ratio. The extent of the enrichment increases with metamorphic rate due to the preferential loss of 14N light nitrogen. Our samples generally have low kerogen carbon nitrogen ratios, and there's no correlation between the bulk carbon nitrogen ratios and the carbon and delta 15N values. Also, the kerogen in our samples yields carbon hydrogen ratio of 2, consistent with a low metamorphic rate equivalent to the predanite pompalite fishes. Moreover, we didn't find a positive correlation between carbon hydrogen ratios and delta 15N kerogen. So all these observations suggest that high delta 15N archived in our samples most likely represent primary signatures. Then we consider potential mechanisms that are responsible for the elevated nitrogen isotopes. The first possibility is denitrification. Basically, this is the process of reduction of dissolved nitrate to nitrogen gas, a process associated with large isotope fractionation. Although sediments deposit in modern oxygen-deficient zones tied to enhanced denitrification often exhibit relatively high delta 15N, but values greater than 15 per mil are rare. Additionally, our phosphorite samples yield negative serial anomalies, indicating well-ventilated water column conditions. I will talk about this later. So this data is inconsistent with the widespread anoxia required to sustain strong denitrification. Also, elevated salinity, as mentioned above, can inhibit microbial nitrogen metabolism, specifically nitrification and denitrification. So taken together, these factors indicate that denitrification alone may not be sufficient to explain the observed delta 15N values. The second possibility is partial nitrification and or partial ammonium assimilation. Both processes can generate isotopically light products, leaving behind residual ammonium enriched in 15N that could be assimilated elsewhere within the same basin. So we would expect a pronounced isotopic heterogeneity that's a wide range of delta 15N values, right? However, our samples yield consistently high 15N values through time and across sites. So ruling out these pathways. Alternatively, we argue that a more parsimonious explanation is ammonia volatilization from water column to the atmosphere. This process imparts a large isotope fractionation and leading to a uniform 15N enriched pool of residual ammonium that can be assimilated into biomass. However, for this mechanism to operate, ammonium must dissociate to hydrogen and ammonia in the water column. The yield of this reaction is governed by high pH. You can see the diagrams here. As a result, elevated delta 15N values, usually greater than 10 per mil, in sediments can be interpreted as a signal of high pH and thus elevated alkalinity. This relationship has been documented in both modern and ancient alkaline lakes systems. So the Phosphoria basin was possibly alkaline. Now we consider the role of alkalinity in the phosphate accumulation. Alkalinity means the strength of buffering acids. Basically, this is the sum of bicarbonate and carbonate ions. Modern alkaline lakes are notable for phosphorus enrichments, in contrast to the widespread phosphorus scarcity typical of most natural waters. This enrichment occurs because in alkaline environments, calcium is preferentially sequestered into carbonated minerals, thereby inhibiting apatite precipitation, and that's a major sink for phosphate. As a result, dissolved phosphate can build up to exceptionally high concentrations, sometimes greater than 15 millimole. By analogy, we propose that elevated salinity in the Phosphoria basin could have promoted phosphate accumulation, a process that may have directly contributed to phosphogenesis. Beyond the role of alkalinity, evaporation would have further facilitated phosphate enrichment in the water bodies. Now we may ask a question on how alkaline the Phosphoria basin was. In fact, alkaline minerals such as trona and nacolite, they are absent from our samples. These minerals are diagnostic indicators of high alkalinity. However, they have extreme solubility and low preservation potential, so the absence does not preclude the possibility of elevated pH. That said, the pH of the Phosphoria basin was perhaps only moderately elevated relative to the coeval global ocean, given that the basin still received seawater influx from open ocean, though somewhat restricted. Importantly, exceptionally high pH and alkalinity are not required to generate high delta 15N in the ammonia pool. In relatively shallow settings, such as the Phosphoria Sea, mixing between water column and atmosphere would have enhanced ammonia degassing. In addition, episodic anoxic conditions would have promoted ammonium buildup, facilitating ammonia loss. Even during intervals with oxic conditions, I will talk about later, elevated salinity would have suppressed microbial nitrification, perhaps allowing ammonium to accumulate in the water column. So collectively, these factors likely contribute to both generation and preservation of the elevated Δ15N signatures. Now, one remaining question is the potential source of alkalinity. The operation of seawater alone cannot account for elevated salinity, so this suggests additional inputs. One possibility is weathering of the surrounding rocks, including Precambrian basement and mafic igneous rocks. Additionally, inflows of carbonate-rich groundwater may have enhanced alkalinity as well. Next, we consider the potential source of phosphorus to the basin. Based on established framework of sequence stratigraphy, you can see the phosphorus-rich intervals correspond to marine transgressions, suggesting that incursions of seawater from open ocean might have replenished phosphate into the basin. This is consistent with previous suggestions, although the basin is semi-restricted. Interestingly, fossil evidence, namely the presence of cool water brachiopods and compounds, further supporting the persistence of marine upwelling. So we also measured rare earth element concentrations in several phosphorite samples. You can see the REE distributions exhibiting a seawater-like pattern, thus ruling out diagenetic alteration and detrital contamination. Importantly, our phosphorus-rich samples are characterized by significant negative stereo anomalies, and these are genuine signatures. Stereo anomalies can shed light on local water column redox dynamics in Earth's history. Don't have time to go through the details, but basically, the negative stereo anomalies suggesting that the phosphorites were deposited under a oxygenated water column. This interpretation is supported by the general association with relatively low TOC and consistent with fossil records of abundant ichneal fauna and benthic fauna. By contrast, samples with low phosphorus contents are characterized by elevated TOC, which may point to deposition under anoxic conditions. So based on this pattern, we propose a two-phase redox phosphorus model. The first phase is characterized by phosphate buildup in the water column and relatively low phosphorus burial in sediments. During episodes of marine transgression, substantial phosphate was supplied through upwelling from open ocean into this basin. Elevated alkalinity combined with evaporation further promoted phosphate accumulation in the water column. This in turn fostered high productivity, marine anoxia, and enhanced organic matter burial. Except the lowermost interval, the water column was likely oxygen deficient but not strongly sulfidic. So under these conditions, phosphorus was gradually sequestered along with organic matter, rather than being recycled back into the water column. However, as organic bound phosphorus continue to settle and become buried, phosphate concentrations in the water column and thus primary productivity would have declined, resulting in a more ventilated water column. At the same time, phosphorus bound to previously buried organic matter could have been recycled and precipitated as apatite within sediments. Interestingly, the carbonate fraction of the phosphorites exhibit relatively negative Δ13C, consistent with this scenario. So this model helps to explain the episodic nature of phosphorus enrichment and may be applicable to other ancient phosphorus-rich successions. Interestingly, available data indicate that many phosphorus are indeed associated with relatively low organic contents. So taken together, our study emphasized potential importance of local factors, specifically alkalinity, evaporation, and basin restriction in driving phosphogenesis within the phosphoria formation and possibly beyond. As I mentioned in the beginning, most phosphatic deposits occur in the Paleoprozoic and Neoprozoic Cambrian transition. So these intervals coincided with episodes of supercontinent breakup, which created numerous rift basins. Weathering of surrounding mafic rocks could have supplied elevated salinity. So if true, our model may help explain the temporal clustering of phosphorized geological record. Thinking more broadly, phosphorus is a fundamental component of cell membranes, genetic material, and cellular energy transfer. So phosphorus is central to the origin and evolution of life. However, in most modern environments, phosphorus is a limiting nutrient. Our results suggest that under specific local conditions, bioavailable phosphate can accumulate in aqueous environments. This has important implications for life's emergence on Earth and potentially on other habitable worlds. Finally, I'd like to thank all my collaborators and special thanks to NASA Postdoctoral Program, NPP, Oak Ridge Associate Universities, or AU, to support my postdoc research, and Duke University as my host institution. Thank you. I'm happy to take any questions.
Thank you so much, Chenyi.
Good. Okay.
Great. Thanks, Mike.
Yeah. Okay. Well, yeah, thanks everyone for tuning into this, and Chenyi for kicking us off. So I'll keep us going on phosphorus. I'm going to take us further back in time and zoom a little out for a broader view of, as I've written here, the causes and consequences of kind of the opposite of what Chenyi was talking about, the scarcity of phosphorus in the global ocean. And here I'm saying in the Precambrian, but I'll mostly focus exclusively on the Archean. So prior to the great oxidation event in the very low oxygen world, what could have caused low phosphorus and what would be the implications of that? And you're seeing a picture here of one of the earliest phosphorites in the rock record from the Zaunega Formation in northwest Russia. It's Paleoproterozoic and comes shortly after the rise of oxygen in the atmosphere. And so it begs the question, sort of like Chenyi was getting at, what are the dominant controls on phosphate burial? Why do we see these phosphate-rich deposits just after the oxygenation of the surface? And so I've got just a few points that I'll touch on here. One is to give us a little more context, that we've gotten some of this already from Chenyi's talk about how and why to think about phosphorus as a limiting nutrient in the biosphere. And then I'll talk about two other things. One, the cause side, a mechanism for low phosphorus in seawater, and then the consequence side. What are the implications of different models for phosphorus scarcity? And I'll note some of this I'm drawing on the somewhat similar talk I gave some years ago for this virtual seminars in Precambrian geology series, which I really enjoyed. I used it as a chance to sort of dive into a review on this topic because I'm interested in a number of facets of it. And so that was 4 years ago now. There's a number of things that have happened since then that I'll try to highlight a bit. But also since then, together with Lewis Alcott, I put together a review chapter for this book, The Archean Earth, about exactly this topic, phosphorus levels and their controls in the Archean. So any of the stuff I'm presenting here, if you're interested in, you could find there sometime in the not too distant future, though the date keeps running away from us a little bit into the future. But hopefully in a few weeks, this will go live online. Okay, so jumping right in. Phosphorus in the biosphere. I'm just going to add a little more context to what Chenyi was saying, shedding light on how I think about this issue, especially in the context of the Archean and the early evolution of the biosphere. And so to frame this, let's think about just the core metabolism that is, life, that is carbon fixation. And so what organisms are doing, primary producers, they're taking inorganic carbon, reacting it with an electron donor or a reductant, to produce reduced carbon, biomass, organic carbon, and then an oxidized byproduct. And so to break this out, what the inescapable parts of this reaction are the inorganic carbon, say CO2 or dissolved inorganic carbon, that becomes organic carbon, biomass. But then there are a number of possible electron donors that organisms could use. And on the early Earth, we think some of these electron donors would have included molecular hydrogen, hydrogen sulfide, reduced iron, and corresponding oxidized byproducts. Now, this can be driven energetically by light if this is phototrophy, so anoxygenic photosynthesis. And we know that organisms have been doing phototrophy, photosynthesis, for much, if not most, of Earth history. I'm showing here just one of the reasons why we know this metabolism's been around, and that is the prevalence of stromatolites, so these sedimentary features that show sediment trapping and growth towards the sunlight source, indicative of phototrophy. This one's from the Death Valley area that's mid-Proterozoic, but we can find these at least 3.5 billion years back, if not earlier in Earth history. So we know that there's been phototrophy going back to the early Earth, certainly through the Archean. This was around, and if you want to then quantify the productivity of an early biosphere, it's plausible that the net rate of this reaction would be limited by the flux of those electron donors to sunlit environments, say the surface ocean. And so estimates of the productivity of an early biosphere that's limited by those reductant fluxes have been generated. Some of the papers doing so are cited at the bottom. But for our purposes here, something really important happened at one point in Earth history, sometime between the formation of the Earth and the Great Oxidation Event. And that is that organisms evolved the ability by coupling to photosystems to use water as the electron donor, split the water molecule and produce organic carbon with a byproduct being now molecular oxygen. And we talk a lot about the molecular oxygen byproduct as an important environmental waste that became what oxygenated the surface, is used for aerobic metabolisms, all these consequences for the evolution of life. But here I want to focus on the use of water as the electron donor, which would then render the biosphere no longer limited by electron donors, so long as you're living, say, in the surface ocean, and would instead usher in an era where we're limited by the next reactant needed for this to move forward, which would be things we call nutrient elements, other constituents of biomass. And so as for when this might have happened, here's our timeline of Earth history. There is still some debate about this. We certainly know that oxygenic photosynthesis was around by the end of, or by the culmination, the Great Oxidation Event, so the latest Archean to earliest Paleoproterozoic. But there's a number of lines of evidence that push that origin back tens, if not hundreds of millions, or even over 1 billion years prior to the GOE. And so from whenever that point is onward, we could expect this nutrient-limited biosphere to prevail. And so let's get us to phosphorus then. If we want to look at this equation in more detail, then we can expand this beyond just the carbon side of biomass production and look at the next two most abundant constituents, nitrogen and phosphorus. And the reason that we focus on phosphorus as this limiting nutrient is that nitrogen available in the atmosphere can be fixed, not by all, but some prokaryotic organisms using the nitrogenase enzyme through nitrogen fixation. And there's no such route to access phosphorus. We're relying on a slow trickle of the chemical weathering of continental crust to bring phosphate into seawater. And so that rate of delivery is thought to limit the total productivity on long timescales of the biosphere. Meaning that from, again, this origin of oxygenic photosynthesis, we could expect a phosphorus-limited biosphere, so long as it resembles more or less primary producers today. And so this is the reason we have such an interest in reconstructing this, the cycling of phosphorus, because if we can do so, we get this first-order constraint on the total possible productivity of the biosphere, which has all sorts of astrobiological implications. So we want to know that, and so let's think now about how phosphorus is cycled through the ocean, starting with the modern, and then we'll tweak that picture for the Archean. And so here's my oversimplified view of how the phosphorus cycle works, and I'll just walk through the salient bits for our purposes, starting with the delivery of phosphorus to the ocean, which here you're seeing on the top left is coming in from rivers, draining the continents, bringing some amount of soluble phosphorus. Much of it is not, in fact, soluble or bioavailable. But that which is dissolved is quickly taken up in the surface ocean by phytoplankton, by primary producers, because of the nutrient-limited nature of primary production. And so that inorganic phosphate shown on the left becomes organic-bound phosphorus. And now that organic-bound phosphorus at some point will sink through the water column and meet one of two fates. It can either get buried in sediments, although what you're seeing is that dashed line is showing this is the exception, and actually the rule is rather that this phosphorus, whether in the water column or once it hits the sediment pile, gets recycled or remineralized back into solution because biomass gets respired by a heterotrophy. And what this does is basically extends the residence time of phosphorus. So here shown on the top, we estimate this on the order of a few tens of thousands of years. and allows us to recycle one phosphate molecule multiple times through the biosphere before it's ultimately buried. So that's why these mineralization and uptake arrows are so much thicker. I should note that there's also some non-zero amount of removal, not just from the organic burial or export production flux, but also from scavenging onto things like iron oxide minerals. And so that's shown here proximal to a hydrothermal source, because today that's the only place where we really see much of this occurring. There's not much iron in seawater otherwise, but proximal to hydrothermal iron inputs, we see scavenging of phosphate onto iron oxides and removal through that pathway. And also, once phosphorus hits sediments, whether with an iron mineral or with organic matter, there's what we call sink switching, where you can liberate phosphate into pore waters and it can enter one of these other phases, say absorbing onto iron minerals there, or also precipitating as authigenic apatite. So to put some numbers on this for us, today the trickle of phosphorus into seawater from rivers is something like 2 teragrams of phosphorus per year, which I just want to highlight is much smaller than the recycled flux that we estimate at something like 1,200 teragrams per year. And so the steady state phosphate concentration, or which is another way of saying that the long residence time, is built up by that recycling. And so if we want to tweak this now and say this is the modern status quo, but let's instead try to think of some ways to drive low phosphate in seawater in the Precambrian or in the Archean specifically. What are some of the parameters we can change here? And there are 2 main parts of this diagram that people have invoked as changing between the Archean and today to give us low phosphate in the Archean. One is that the prevalence of this iron scavenging flux is thought to potentially have been much higher. We know there's voluminous iron formation deposition. We'll look at this more in just a moment. And so presumably the scavenging of phosphate onto those iron minerals, whether they're iron oxides or even actually reduced or mixed valence iron minerals, can all, we have now seen, scavenge iron from solution. So regardless of primary mineralogy, we could expect some amount of that phosphate scavenging in a more iron-rich reducing ocean. In addition to that, not mutually exclusive, before you could even have the phosphate ions released, available to be scavenged onto iron minerals, it's possible that recycling flux was cut off. And this would be because in the low oxygen, but also low oxidant, so low sulfate and nitrate and other oxidized species, low oxidant Archean ocean, you wouldn't have the oxidizing power to oxidize your biomass and release the nutrient content. You'd instead bury the organic matter efficiently in sediments and together with it, its nutrient content. And so either or both of these mechanisms could contribute to phosphorus scarcity in the Archean. I've broken them down here for sake of argument into the iron scavenging camp, the limited recycling camp. There are a number of studies shown here that touch on different aspects of this. And what I'm going to do first here is just look at some ways of trying to look for evidence for one or the other of these mechanisms at play. And I'm going to focus on the delivery of phosphate to sediments because they make different end member predictions. On the left, the iron scavenging model would say that iron is being delivered to sediments with phosphorus with iron. Whereas on the limited recycling model, we'd think that phosphorus is supplied to sediments mostly with organic matter. And so let's look for evidence of either of those mechanisms at play. And I'm going to start, mainly focusing on the settings where you'd really expect iron scavenging to show up, which are iron formations, right? If we had any of this occurring anywhere, we would expect that these super iron-rich deposits would show evidence stoichiometrically of phosphorus delivery to sediments with the iron. And there's a bunch of data we can use to test this in different ways. So I'm going to start with some of the earliest data that sort of set this iron scavenging idea off in motion from Burim and Canfield in 2002. They're here looking at the phosphorus content and phosphorus to iron ratio of Archean and early Paleoproterozoic iron formations. And let's zoom in on this one here. This is the Dales Gorge iron formation. So this is in the very earliest Paleoproterozoic of Australia prior to the GOE. What we're going to look at is on the y-axis, this is cast as a metal flux per year, but this is based on the concentrations observed of phosphorus, but also of other trace elements in that unit, and then converted into a flux using estimates of depositional rates of the Dales Gorge member. And we're going to try to match not just the amount of phosphorus, but of all of the suite of trace elements with different sources. One being adsorption onto ferrihydrite, so an iron oxide source scavenging like we see happening proximal to hydrothermal systems today, and another being export production, phosphorus and these other trace elements coming to the sediment with organic matter. And so, like I said, measurements were made of the Dales Gorge member. So we have the concentration data that are converted to the flux on the y-axis. That's what the red lines are showing you here. So the amount of supply of phosphorus, but also zinc, cobalt, and so on needed to give rise to the observed composition of the Dales Gorge member. And then experimentally, the export production profile was estimated with cultures of cyanobacterial and photoferrotroph biomass, whereas on the right, the inorganic experiments of adsorption onto ferrihydrates were aimed to meet those fluxes. And what we see actually, so in this work from Kurt Konhauser's group, is that in both models, you can match the desired amount of phosphorus. You can get that supply to match the depositional rates, we think. But you don't match the rest of the trace element profile if you have the ferrihydrate source, whereas you do much better if you have the organic matter source. And so this would suggest that despite the prevalence of iron and the lack today of organic matter in these iron formations, we think the original supply to the sediments was more likely the phosphorus coming with the organic matter. That's one way to think of this. We can do it another way, and I'll, in this case, show data from the Kuruman iron formations. Now we're still in Paleoproterozoic, moving over to South Africa. Here we're going to look at phosphorus just versus iron concentration, looking for a correlation here. We would think if that was the dominant supply vector, and similarly in the Griquaton iron formation, similar Paleoproterozoic of South Africa. If we look at the phosphorus versus iron concentrations, we see no correlation, which would basically is saying to us the amount of phosphorus does not depend on the amount of iron in the sample. And instead, we could say, well, do we see such a correlation if we look at phosphorus versus organic matter? Though we have this issue that I mentioned before, that while today there's very little organic matter, we think that there may have been substantial organic matter exported to the sediments, and it's been oxidized, namely through a dissimilatory iron reduction. And so you can estimate what I'm showing on the x-axis here as original TOC using this equation up top, where you're taking the amount that's observed today, it's a very small amount, and then adding to that this correction that comes from the observation of the inorganic carbon fraction. So there's siderite iron carbonate in these rocks that has isotopically negative, so very low isotopically light carbon that is derived, we think, from the respiration of organic matter. And we can, by assuming a composition isotopically of that primary organic matter, solve for how much has now been converted to siderite and reconstruct the original TOC. And if you do that, we see that in both cases, well, on the left, in the case where you have more data in the Kuruman, you see actually some correlation between phosphorus and reconstructed TOC. But in both cases, you can see these C to P ratio lines. All of the data fall well below the modern likely C to P ratio, even below a C to P of higher than modern, which is potentially the case for early cyanobacteria, suggesting that we need no other supply mechanism besides the organic matter. In other words, there's no extra phosphorus that we can't explain except unless we lean on a biomass source. And so based on this approach and also the experimental one from Konhauser et al., in these cases, despite there being little organic matter today, it looks like we see stoichiometric evidence of phosphorus supply with organics rather than with iron minerals, which would support that limiting recycling model, and especially important to keep in mind in these iron-rich depositional settings. I'll note, though, that of course I was happy with that aligning with these model predictions some years ago, and in the meantime, some other work has now shown that there are places where you see the opposite, where you can find in these Neo-Archean carbonates of South America, a very strong correlation of phosphorus with iron, which is what we would expect if iron scavenging were important. And not only that, but we see that there's a much shallower slope in the Archean samples, phosphorus versus iron, than in modern hydrothermal sediments, which is consistent with lower phosphate levels in Archean seawater. And so if we're coming back to trying to judiciate between these two models at play. I don't think there's such a thing as an ocean that was entirely at one end member or the other. There could be spatial variability and or temporal variability, say, as a function of hydrothermal iron fluxes. But what I want to do from here is say that if we take the limited recycling model as playing some role, not only because of these indications of transport to sediments with organics, but what we clearly know about the scarcity of oxidants in Precambrian seawater, we can then consider the implications and the feedbacks that we would encounter as we oxidized Earth's surface and relieved that limitation on phosphorus recycling. And so that's what I want to focus on now, are the implications of this limited recycling model, namely just one aspect of this that I think is really neat, and that is the role of sulfate as a regulator of Precambrian phosphorus burial, or on the flip side, of its recycling. And so, like I've been saying throughout the talk, there's, we think, a scarcity of oxidants, not just O2, but of all oxidizing power in Precambrian seawater, especially in the Archean. Here is a compilation that we put together some years ago of the main electron acceptors used for biomass recycling for remineralization, and their abundance estimated through time from a variety of studies. So on the right-hand side, we see today, note that this is a log axis spanning many log units. We have quite a bit of sulfate, 28 millimoles per liter in modern seawater. And also, if we equilibrate surface waters with our oxygenated atmosphere, we fit a few hundred micromoles per liter of O2. Together, that's plenty of oxidizing power to respire, recycle all biomass that's sinking, far, far excess of sulfate and even in excess of oxygen. But as we go back through time, all of these, we see our constraints suggest orders of magnitude lower concentrations, both in sulfate and oxygen, and especially in the Archean, where we think there is vanishingly low dissolved oxygen, except very localized oxygen oases. We would rely heavily on sulfate for oxidizing power to drive organic matter remineralization. And I'd note here that iron box, that's meant to represent ferric iron available for dissimilatory iron reduction. That's a very conservative upper estimate. It's upper limit. It's assuming that our upper limit on ferrous iron concentrations that comes from iron mineral solubility could be quantitatively converted to ferric iron and then entirely used for organic matter respiration. And so that's a set of conservative assumptions, and I think we would expect lower than that amount of iron. So sulfate really would have a lot of work to do to carry the load of recycling phosphorus back to the water column. And not only that, but I'm showing in this modified version of the cartoon from earlier, there's a second aspect to the role of sulfate in phosphorus recycling to the water column. The one is shown on the left, that the transport of organic bound phosphorus to sediments, even when there's no O2, if you have sulfate, you can respire the biomass, let the phosphorus back to the water column. But then also the byproduct of that sulfate reduction, hydrogen sulfide, will tend to react with iron in sediments, producing iron sulfides. And importantly, iron sulfides do not absorb phosphate, whereas the iron oxides would. And so that's another way of cutting off the iron trap. And collectively, what that means is there's more phosphate that can escape back to the ocean. So the growth of the sulfate reservoir would allow this, but in a low sulfate world, we would expect very little of that recycling to occur. And we can actually see this if we look in modern analogs. I think this is really neat. This work from Carrico and others in, I think, 1988, they looked at anoxic sediments as a function of sulfate, which we can't find any low sulfate analogs in the modern ocean. But if you go to non-marine settings, so they're looking at anoxic lakes, you can find anoxia, but low sulfate. And on the y-axis, relative P release, again, note this is a log scale, they see a steep drop-off in the amount of phosphorus recycled to the overlying water column when they get to low sulfate, but still anoxic conditions, meaning it's staying buried with the biomass as we would sort of predict in this model. So I wanted to look at the implications of that relationship, and I did it in a pretty simple way. I just took that estimate of phosphorus release versus sulfate, cast that as a C to P ratio. In other words, saying that you start with a C to P ratio of something like today, the Redfield ratio, 106:1. If you recycle phosphorus back to sediments while you respire biomass in sediments and you don't let it get trapped as apatite or bound to iron, then you'll have a higher C to P ratio in those sediments. That means the phosphorus is now back in the water column. And so I've taken that relative recycling, cast it as a C to P ratio, where the higher C to P occurs in the sulfate-rich settings, but then there's preservation of the phosphorus with the organics at the low sulfate. And I just cast this here with these this Monod expression with this half saturation constant K term using different ranges from the literature. That's what you see as these dashed lines fitting that data. And basically then wanted to ask the question, if this relationship exists, then by changing the amount of sulfate, you can change the amount of sulfate recycling back to the water column, which will change productivity. It'll change the amount of oxygen in the atmosphere, delivery of sulfate through oxidative weathering, and you might get positive feedbacks as you grow the sulfate reservoir in the late Archean or early Paleoproterozoic. And so I just last want to show these model outputs exploring that feedback, and I'm doing it in two scenarios, what I'm calling a whiff of oxygen type of event on the left or a GOE type of event on the right. They're both set off in the exact same way. So on the top panel, I take a baseline phosphorus delivery flux and then give a pulse of extra phosphorus coming in from rivers. It could be for a variety of reasons, doesn't matter here. And the only difference between these two scenarios is that the GOE event here gets a little extra pulse relative to the whiff event. What that does, the pulse of phosphorus, if we have a phosphorus-limited biosphere, causes more productivity, organic carbon burial, oxygen accumulation. So if you look in the bottom panels, we get from this vanishingly low baseline a small pulse of atmospheric oxygen. These are units of PAL, present atmospheric levels. But I'm looking at two different cases for each of these events. In the blue lines, I'm arbitrarily saying there's no such thing as this phosphorus recycling. So I'm just holding constant C to P ratios. And that's what the third panels are showing, a flat line in C to P. So there's no preferential recycling when, like we know, is driven by sulfate, let's say in anoxic settings. And then in the red dashed line, we have that sulfate driven recycling in the model. And what you see is that if we allow that to be in the model, in the red dashed lines, the C to P ratio will go up during the event. And that also means that we get more, so there's more phosphorus in the water column, more productivity. The PO2 pulse gets larger, and that oxygen in the atmosphere is driving oxidative weathering, bringing sulfate to seawater. And so we see the sulfate pulse occurring as well. So this would either mean that in the case of a whiff of oxygen, this feedback is giving you a stronger whiff because the sulfate delivered through oxidative weathering gave you more phosphorus recycling, more productivity. Or what I think is most neat is that in the GOE type event, that feedback is the difference between a whiff and pushing you to this runaway accumulation of oxygen to the point where you're buffered then by oxidative weathering, which is by definition what we would consider the GOE. And so I think this is a feedback that would push us towards the GOE occurring. It would be inescapable as you grow the sulfate reservoir and start oxygenic photosynthesis. And I should note, this is a simple model result that's showing us a type of feedback in a little, sort of toy model calculation, but there's some data now. These are two papers in the last few years showing in using C to P ratios and phosphorus speciation analysis that we see evidence actually of during these transient oxygenation events in the latest Archean, signs of that phosphorus recycling kicking on. So I think there's something to this. And I think the implications for the role of sulfate in the phosphorus cycle are really neat. So just to wrap that up, just a few main conclusions. I think that the scarcity of oxidants in Archean or just more generally Precambrian seawater would have created this throttle on phosphorus recycling. Doesn't mean there was no iron scavenging ever occurring, but it does mean that mode of limitation has these neat implications or sensitivities, namely as it relates to sulfate delivery. And so I think there would be this positive feedback where the oxidative weathering, when we start to accumulate oxygen, brings more sulfate and allows more recycling and oxygen production and so on. And linking it back to Chenyi's talk, it makes me wonder about the secular distribution of phosphorites through Earth history, how much that might be related to growth of the sulfate reservoir and the difference in timescales and spatial representation of that versus just oxygen accumulating in surface environments. So with that, I will just throw up some acknowledgements and happy to take questions. Thanks again.